Welcome to Sleeping Researcher, where we explore the wonders of science, guiding you gently into a state of relaxation and tranquility. This is the fourth video, and I am seeing some unique viewers trickle in. I would like to extend my gratitude to people listening to my narrations. In this episode, we will embark on a fascinating journey through the intricate world of DNA, the essence of life itself, and its profound connection to evolution and synthetic biology. As we delve into these interconnected realms, we'll uncover the remarkable story of how life on Earth has transformed over millions of years, shaped by the delicate dance of genetic information passed through generations. DNA, with its elegant double helix structure, serves as the blueprint for all living organisms, holding secrets that scientists continue to unravel. We will also explore the exciting advancements in synthetic biology, where researchers harness the power of DNA to create new life forms and innovative solutions to some of our greatest challenges. This intersection of natural evolution and human ingenuity opens up a world of possibilities, inviting us to ponder the future of life itself. So, settle in, take a deep breath, and allow your mind to drift as we explore the remarkable connections between DNA, evolution, and synthetic biology. Let's begin our journey into this awe-inspiring scientific realm together. Imagine a world where the intricate dance of life unfolds in the tiniest of particles, where each movement is choreographed by a remarkable molecule known as DNA. This double helix structure, often described as the blueprint of life, holds the secrets to our existence. Encoded within its spiraling strands are the instructions that guide the development, functioning, growth, and reproduction of all living organisms. From the simplest bacteria to the most complex mammals, DNA serves as the foundational element of biological processes, ensuring that life continues in its myriad forms. As we delve into this fascinating world, we uncover not just the essence of what makes us unique, but also the shared heritage that connects all forms of life on Earth. It is through understanding DNA that we can begin to appreciate the profound intricacies of life itself. At the core of DNA's significance lies its role in evolution, the process by which species adapt and change over generations is intricately tied to the genetic variations encoded within the DNA of each organism. Mutations, which can occur naturally during DNA replication, introduce new traits that may offer advantages in survival or reproduction. Over time, these traits can become widespread within a population, leading to the emergence of new species. This dynamic interplay between genetic variation and environmental pressures drives the evolution of life. As we explore the mechanisms of evolution, we begin to see how DNA acts not only as a repository of information, but also as a catalyst for change, shaping the diversity of life we observe today. Synthetic biology represents a fascinating intersection of our understanding of DNA and the potential for innovation. By manipulating genetic material, scientists can design and construct new biological parts, devices, and systems that do not exist in the natural world. This field holds promise for a wide range of applications, from creating biofuels and pharmaceuticals to developing crops that can withstand extreme environmental conditions. Through synthetic biology, we are not merely passive observers of nature's process. We become active participants, rewriting the code of life itself. As we harness the power of DNA, we must also tread carefully as ethical considerations arise regarding the implications of our modifications. This delicate balance between innovation and responsibility will shape the future of biology. Understanding the structure and function of DNA is foundational for both evolutionary biology and synthetic applications. As we uncover the layers of complexity within DNA, we also recognize its potential as a tool for solving some of the world's pressing challenges. From medical advancements to environmental sustainability, the possibilities are vast. Each discovery in the realm of DNA not only enriches our knowledge, but also deepens our connection to the natural world. This journey through the microscopic realm of life invites us to reflect on our place within the tapestry of existence. In this context, we stand on the precipice of a new era in which the secrets of life are not merely to be observed, but can be actively engaged with and reshaped. As we delve deeper into the realms of evolution and synthetic biology, we will uncover the profound implications of our newfound capabilities. The journey ahead invites us to explore how our understanding of DNA can lead to transformative changes in the way we approach health, sustainability, and the very fabric of life itself. The intricate structure of DNA, often described as a double helix, is a marvel of nature, elegantly designed to house the genetic information essential for life. Composed of two long strands that coil around each other, this structure is formed by sequences of nucleotides, which serve as the building blocks of genetic material. Each nucleotide consists of a sugar, a phosphate group, and a nitrogenous base. The arrangement of these bases, adenine, thymine, cytosine, and guanine, encodes the instructions for building and maintaining all living organisms. This remarkable arrangement not only enables the storage of vast amounts of information, but also facilitates the replication process that is vital for cellular division and inheritance. Each time a cell divides, the double helix unwinds, and the strands serve as templates to recreate identical copies of genetic material, ensuring continuity across generations. The elegance of DNA's double helix is not merely structural, but also functional, revealing insights into the mechanisms of evolution. Variations in the sequences of nucleotides can lead to differences in traits among organisms, a principle that underpins the theory of natural selection. Over long periods, these subtle changes accumulate, allowing populations to adapt to their environments and leading to the rich diversity of life we observe today. The study of DNA enables us to trace the lineage of species, illuminating the connections and divergences that have shaped the tree of life. By understanding the genetic blueprints of various organisms, we can appreciate the intricate web of relationships that bind us all. In recent years, advances in synthetic biology have begun to harness the principles of DNA structure and function for innovative applications. Scientists are now capable of designing and constructing new biological parts, devices, and systems that can perform specific functions. This burgeoning field holds promise for addressing critical issues such as disease treatment, biofuels, and environmental remediation. By synthetically manipulating DNA, researchers can create organisms with tailored characteristics, enabling breakthroughs that were once thought to be the realm of science fiction. The potential for synthetic biology to enhance our lives is immense. Yet, it also beckons us to contemplate the ethical implications of such powerful technologies. As we delve deeper into the realm of DNA and its synthetic counterparts, it becomes evident that our understanding of this molecular structure is not only crucial for evolutionary biology, but also for the future of biotechnology. The ability to decode and manipulate genetic information offers a pathway towards solutions that could significantly improve health outcomes and environmental resilience. Each step forward in our comprehension of DNA and its applications invites us to reflect on the responsibilities that accompany such knowledge. The intricate dance of life, encoded within the double helix, serves as a reminder of the delicate balance we must maintain with the natural world. The essence of heredity lies within the coils of DNA, where a sequence of nucleotides encodes the instructions for building and maintaining an organism. This molecular blueprint is not merely a static record, but a dynamic agent that guides the expression of traits across generations. Each parent contributes half of their genetic material, creating a unique combination in their offspring, a concept celebrated in the study of inheritance patterns. The intricacies of this process reveal the underlying mechanisms that drive evolution, as variations in DNA can lead to different phenotypes, influencing survival and reproduction in the ever-changing tapestry of life. As we explore the implications of DNA in heredity, we find ourselves at the intersection of evolution and synthetic biology. Evolution acts as a sculptor, shaping species over eons through the gradual accumulation of genetic changes driven by environmental pressures. These adaptations are encoded in DNA, illustrating how life continually evolves in response to its surroundings. In contrast, synthetic biology empowers us to intentionally manipulate these genetic codes, crafting new forms of life with specific characteristics. This interplay of natural and synthetic evolution raises profound questions about our role as stewards of genetic information. The ability to synthesize DNA and create novel organisms offers a lens through which we can examine both the potential and the ethical considerations of genetic manipulation. By understanding the principles of heredity, we can harness the power of DNA to innovate solutions for pressing issues such as food security, disease resistance, and environmental sustainability. However, with this power comes the responsibility to consider the long-term implications of our actions. Just as natural selection operates in the wild, our decisions in the laboratory can echo through ecosystems and societies. The journey of DNA from a simple molecule to a complex system of heredity reflects the profound interconnectedness of life itself. Each strand of DNA tells a story of survival, adaptation, and continuity, serving as a reminder of the delicate web that binds all living organisms. As we harness the capabilities of synthetic biology, we must remain mindful of the lessons gleaned from natural evolution. The potential to create and modify life should compel us to tread carefully, ensuring that our innovations align with the principles of sustainability and respect for the natural world. At the heart of evolutionary processes lies the mechanism of mutation, a natural phenomenon that fuels the diversity of life. Mutations are changes that occur in the DNA sequence, arising from various factors such as environmental influences, replication errors, or even spontaneous chemical reactions. While some mutations may be neutral or even detrimental, others can confer advantageous traits that increase an organism's chances of survival in a changing environment. This genetic variability is essential, as it provides the raw material for evolution, enabling populations to adapt over generations. The delicate balance between stability and change is what allows species to thrive in their respective niches. As we delve deeper into the intricacies of DNA, we begin to appreciate the elegance with which nature orchestrates these changes, reminding us that each mutation is not merely a random event, but rather a potential catalyst for innovation and resilience. The significance of mutations extends far beyond mere survival. They are the foundation upon which the incredible tapestry of life is woven. Over eons, these small alterations in DNA have led to the emergence of new traits, behaviors, and even entirely new species. This gradual process of adaptation and natural selection underscores the principle that life is not static, but rather a dynamic interplay of genetic possibilities. Each organism, shaped by its unique genetic code, embodies the history of countless mutations that have been sifted through the rigorous test of survival. By examining the evolutionary tree, we can trace the lineage of species and observe how mutations have facilitated the rise of complexity and diversity in the natural world. This rich history serves as a reminder that change is a constant and necessary component of life. The field of synthetic biology invites us to engage with the concept of mutation in new and profound ways. By understanding the mechanisms that govern genetic change, scientists are now capable of designing and constructing new biological systems. This innovative approach holds the promise of addressing some of the most pressing challenges facing humanity, such as food security, disease management, and environmental sustainability. However, as we explore the potential of engineered organisms, we must remain cognizant of the ethical considerations that accompany such powerful tools. The lessons learned from natural evolution remind us that every alteration carries consequences, and the responsibility lies with us to wield this knowledge with care. The intricate dance between natural mutations and human ingenuity calls for a careful balance, ensuring that our endeavors respect the integrity of existing ecosystems. In the grand narrative of life, it becomes evident that mutations are not merely anomalies, but essential drivers of evolution. They embody the spirit of exploration that characterizes living organisms, constantly seeking new pathways to adapt and flourish. As we stand at the crossroads of natural evolution and synthetic innovation, we are presented with a unique opportunity to reflect on the interconnectedness of all life forms. This perspective compels us to consider how our creations might align with the wisdom of nature, fostering a harmonious relationship between technology and the environment. With each step forward in synthetic biology, we are reminded that the journey of evolution is ongoing, and our role in it is both a privilege and a responsibility. Natural selection is a profound mechanism that intricately weaves the fabric of life through the variations found in DNA. Within the double helix structure of DNA lies the blueprint for every organism, coded in sequences of nucleotides that dictate not only physical traits, but also behavioral tendencies. When mutations occur, they introduce new traits into a population, some of which may enhance an organism's ability to survive in its environment. Over time, those traits that offer a competitive advantage become more prevalent, as individuals with these advantageous characteristics are more likely to thrive and reproduce. This natural process, guided by environmental pressures, shapes the evolutionary trajectory of species, demonstrating that life is not static, but a dynamic tapestry of adaptations. As we delve deeper into this intricate dance of genetics and survival, we begin to appreciate the elegance of natural selection and its role in fostering diversity among living organisms. The interplay between DNA variation and natural selection provides a rich ground for understanding the evolution of life on Earth. Each generation reflects the culmination of countless adaptations, each a testament to the resilience of life in the face of change. Consider the various traits that have evolved in response to environmental challenges, be it the camouflage of a chameleon, the speed of a cheetah, or the resistance of certain plants to drought. These traits are not arbitrary. They are the result of countless interactions between organisms and their surroundings, sculpted by time. As we observe these processes, we are reminded of the delicate balance that sustains ecosystems, and how each species plays a vital role within this intricate web of life. Herein lies the beauty of evolution, a continuous journey shaped by the forces of nature, where each twist and turn opens new avenues for exploration. In our exploration of synthetic biology, we find ourselves at a remarkable intersection of natural selection and human ingenuity. By harnessing the principles of DNA, scientists are now able to engineer organisms with desired traits, effectively guiding their evolutionary paths. This innovative approach allows us to address pressing challenges such as food security, disease resistance, and environmental sustainability. Yet, as we embark on this bold venture, we must remain mindful of the lessons imparted by natural selection. The wisdom of millions of years of evolution teaches us that every action in the biosphere has consequences, and the introduction of synthetic organisms into natural ecosystems necessitates careful consideration. We are reminded that while we possess the power to create, we must approach this responsibility with respect for the intricate balance that nature has perfected over eons. As we continue to navigate the complexities of synthetic biology, it becomes increasingly clear that our understanding of natural selection can guide us in our endeavors. By mimicking the processes that have driven evolution, we can create organisms that not only serve human needs, but also benefit the environment. For instance, through the development of bioengineered crops that require fewer resources or produce higher yields, we can support a growing population while minimizing our ecological footprint. The potential of synthetic biology is vast, yet it is intertwined with the fundamental laws of life as shaped by natural selection. Our challenge lies in ensuring that our innovations align with the rhythms of nature, fostering a synergistic relationship that honors both scientific discovery and ecological integrity. The intricate relationships among various species can be visualized as a vast and sprawling tree, often referred to as the tree of life. Each branch represents a lineage, showcasing the evolutionary pathways that have emerged over millions of years. The roots of this tree delve deep into the ancient past, where the earliest forms of life began their journey on this planet. Through the study of DNA, we can unravel these connections, revealing how diverse life forms are interrelated. By examining the genetic material shared among species, scientists are able to trace back the lineage of all living organisms, illuminating the common ancestry that binds us together. This genetic tapestry not only illustrates the evolutionary process, but also emphasizes the remarkable adaptability of life. As species evolved, their DNA underwent changes driven by the forces of natural selection, mutation, and genetic drift. Each genetic variation, while seemingly small, contributes to the vast diversity we observe today. As we delve deeper into the world of synthetic biology, it becomes evident that our understanding of DNA and its evolutionary significance can inform our innovations. By studying the genetic blueprints of organisms, scientists can identify key traits that have allowed certain species to thrive in specific environments. This knowledge can be harnessed to engineer new organisms that possess desirable characteristics, such as resilience to disease or enhanced nutritional value. For instance, researchers are now able to design bacteria that can break down pollutants, thereby aiding in environmental restoration efforts. The interplay between synthetic biology and evolutionary principles is a testament to the power of nature as a guide. By respecting the evolutionary context from which these organisms arise, we ensure that our creations are not only effective, but also harmonious with the ecosystems they inhabit. The study of the tree of life also sheds light on the ethical considerations surrounding synthetic biology. As we manipulate genetic material, we must remain mindful of the interconnectedness of species and the potential consequences of our actions. Each modification we make could ripple through the ecosystem, affecting not just the target organism, but also its relatives and the environment at large. This awareness encourages a sense of responsibility, urging us to approach synthetic biology with caution and respect for the delicate balance of life. Furthermore, as we explore the genetic relationships among species, we are reminded of the importance of preserving biodiversity. The loss of a single species can have far-reaching effects, underscoring the need to protect and conserve the rich tapestry of life that has evolved over millennia. In essence, the tree of life serves as a reminder of the beauty and complexity of evolution. It illustrates not only how life has adapted and diversified, but also how all organisms are connected through a shared history. This profound understanding can guide our efforts in synthetic biology, allowing us to create solutions that are not only innovative, but also sustainable. As we continue to explore the potential of engineered organisms, let us remain anchored in the principles that have governed life on Earth for eons. By prioritizing the health of our ecosystems and the integrity of natural processes, we can ensure that our advancements contribute positively to the world around us. In essence, the interplay between natural selection and synthetic biology poses both opportunities and challenges. As we harness the power of genetic engineering, we must remain mindful of the lessons imparted by nature's own evolutionary processes. By fostering a deep understanding of how advantageous traits arise and persist, we can better navigate the complexities of ecological interactions, ensuring that our innovations are in harmony with the natural world. The modern evolutionary synthesis represents a harmonious convergence of Darwinian thought and the burgeoning field of genetics. This synthesis emerged from the realization that the mechanisms of heredity, first elucidated by Gregor Mendel, could seamlessly integrate with Darwin's theory of natural selection. In understanding the interplay between genes and the environment, we gain insights into how variations within populations arise and persist. Genetic mutations, whether subtle or significant, can confer advantages or disadvantages in a given environment, ultimately influencing the survival of species. This intricate dance of chance and necessity is not merely a theoretical construct, it is a living process, observable in nature and increasingly reproducible in laboratory settings. As we delve deeper into the tenets of the modern evolutionary synthesis, we uncover the profound implications for our understanding of biodiversity. Each species, shaped by the forces of natural selection acting on genetic variation, carries within its DNA the story of its evolution. This genetic narrative is a testament to the resilience and adaptability of life, revealing patterns that guide us in appreciating not only the diversity of life forms, but also their interconnectedness. The more we learn about the genetic underpinnings of these processes, the better equipped we become to engage with the challenges posed by a rapidly changing world. The principles embedded within the modern evolutionary synthesis also provide a framework for synthetic biology, a field that seeks to redesign and manipulate biological systems. By leveraging our understanding of genetic mechanisms, scientists are now able to engineer organisms with novel traits, pushing the boundaries of what is possible in biotechnology. This endeavor is not without ethical considerations, as we tread carefully to respect the delicate balance of ecosystems and the intrinsic value of existing life forms. Thus, as we embark on this journey of innovation, we must remain cognizant of the lessons learned from evolution, ensuring that our synthetic creations harmonize with the rhythms of nature. Furthermore, the modern evolutionary synthesis invites us to ponder the future trajectory of life itself. As we manipulate genetic codes and create new organisms, we must ask ourselves how these synthetic life forms will interact with the existing tapestry of life. Will they enhance resilience in ecosystems, or will they inadvertently disrupt age-old relationships? The answers lie in our ability to apply the wisdom of evolutionary theory to our scientific pursuits, fostering an approach that is as much about preservation as it is about innovation. In navigating these complex intersections of evolution, genetics, and synthetic biology, we are reminded of the enduring legacy of the modern evolutionary synthesis. Our understanding of how life evolves is not just a collection of facts. It is a dynamic, ongoing narrative that shapes our relationship with the natural world. As we continue to explore the potential for engineered organisms, let us hold steadfast to the principles that have guided life on Earth, ensuring that our endeavors contribute to a sustainable future for all living beings. The intricate dance of molecules that forms the essence of life is captured within the double helix of DNA. This remarkable structure, a sequence of nucleotides, encodes the very instructions that allow organisms to grow, develop, and adapt to their environments. Each twist and turn of the helix is a testament to millions of years of evolution, where natural selection has sculpted these genetic blueprints into the diverse tapestry of life we observe today. As we delve deeper into the realm of synthetic biology, we find ourselves at the intersection of natural processes and human ingenuity. Here, we harness our understanding of DNA to design and construct new biological parts, tools that can serve various purposes, from addressing environmental challenges to advancing medical therapies. This endeavor is not merely an act of creation, it is a profound responsibility. As we wield this power, we must remain attuned to the complexities of ecosystems and the delicate balance that sustains life on our planet. In the landscape of synthetic biology, the ability to manipulate genetic material allows scientists to engineer organisms with precision and intent. By introducing specific genes or altering existing ones, we can guide the evolutionary process in ways that were once unimaginable. This practice is reminiscent of selective breeding, yet it operates on a molecular level, enabling us to produce organisms with traits tailored to our needs. For instance, we can create bacteria that consume pollutants, thereby assisting in the remediation of contaminated environments. Such innovations highlight the transformative potential of synthetic biology, merging the wisdom gleaned from evolution with the tools of modern engineering. However, as we embark on these pioneering journeys, we must remain vigilant stewards of both ethics and ecology. The implications of our designs extend beyond laboratory walls, influencing the very fabric of ecosystems and the myriad forms of life they support. The principles of synthetic biology not only offer solutions to pressing global issues, but also challenge our understanding of what it means to be alive. By creating organisms with novel functionalities, we blur the lines between natural and artificial life, prompting profound questions about identity and agency in the biological realm. The debate surrounding the ethical implications of these creations is as complex as the science itself, invoking discussions about biodiversity, genetic ownership, and the moral responsibilities of scientists. As we navigate this uncharted territory, we must keep in mind the lessons learned from the study of evolution, resilience, adaptability, and interdependence. These qualities remind us that our innovations must harmonize with the rhythms of nature, fostering a relationship that is both respectful and reciprocal. As we stand on the threshold of this new era, the potential for synthetic biology to revolutionize medicine is particularly promising. Imagine microorganisms engineered to produce life-saving drugs, or modified plants that yield enhanced nutritional content. Such advancements could address some of the most critical health and food security challenges facing humanity. Yet, with such promise comes the imperative to ensure safety and efficacy through rigorous testing and ethical oversight. The integration of synthetic biology into our health systems requires a collaborative approach, bridging the gaps between scientists, ethicists, policymakers, and the public. By fostering open dialogues and transparent practices, we can cultivate a shared vision for the future of biotechnology, one that prioritizes well-being and sustainability. In this evolving narrative of life, the story of synthetic biology serves as a reminder of our interconnectedness with the natural world. As we strive to innovate and improve the human condition through engineered organisms, we must remain anchored in the principles of evolution and ecology. Our journey is not solely about creating. It is also about understanding the implications of our creations and their impact on the world. As we proceed forward, let us embrace the complexity of these challenges with humility and foresight, ensuring that our efforts contribute to a thriving biosphere for all living beings. In the intricate dance of DNA and evolution, synthetic biology emerges as a powerful tool, allowing us to harness the principles of life itself. By manipulating the building blocks of life, we can engineer organisms that serve specific purposes, addressing some of our most pressing global challenges. For example, the production of biofuels from engineered microorganisms offers a promising pathway to reduce our reliance on fossil fuels and mitigate climate change. These organisms can convert waste materials into energy-rich compounds, presenting a sustainable alternative that aligns with the natural cycles of life. As we delve deeper into this realm, we find ourselves not only creating, but also learning from nature's wisdom, as evolution has already optimized countless processes over millennia. Beyond biofuels, the applications of synthetic biology extend into the realm of pharmaceuticals, where the synthesis of complex molecules can lead to groundbreaking treatments for diseases. By programming bacteria or yeast to produce therapeutic compounds, we can streamline drug development, reduce production costs, and enhance accessibility for patients around the world. This innovative approach not only accelerates the discovery of new medicines, but also allows for the customization of treatments tailored to individual genetic profiles, embodying the essence of personalized medicine. Such advancements remind us of the interconnectedness of our endeavors, as each engineered organism carries the potential to impact human health and well-being significantly. As we explore the vast landscape of synthetic biology, it is crucial to remain mindful of the ethical and ecological implications of our innovations. Each new organism we create is not merely a tool, but a living entity that interacts with its environment in complex ways. The potential for unintended consequences necessitates rigorous testing and a deep understanding of the ecosystems we aim to influence. This awareness fosters a sense of responsibility, urging us to consider the long-term effects of our creations on biodiversity and ecological balance. By engaging with the principles of evolution, we can navigate these challenges with a commitment to sustainability and harmony. Furthermore, synthetic biology holds the promise of revolutionizing agriculture through the development of crops that can withstand climate change and reduce the need for chemical inputs. By engineering plants to enhance their resilience or nutritional content, we can address food security while conserving natural resources. This innovative approach not only supports the growing global population, but also aligns with the principles of ecological stewardship, ensuring that agricultural practices contribute positively to the environment. As we continue to explore these possibilities, we remain ever mindful of the intricate web of life that sustains us. In this ongoing exploration of synthetic biology, we find ourselves at the intersection of technology and nature, where the potential for innovation is limited only by our imagination. As we engineer life forms to solve the pressing issues of our time, we must also cultivate a deep respect for the natural processes that have shaped our planet. Striking a balance between innovation and conservation is essential. It is through this lens that we can appreciate the beauty and complexity of life, while creating solutions that benefit all. The discovery of DNA, the molecule that carries the genetic blueprint of all living organisms, has fundamentally altered our understanding of biology. Each twist and turn of its double helix structure tells a story of life, evolution, and the intricate dance of genes. Through the process of natural selection, organisms have adapted and evolved over millions of years, crafting a diverse tapestry of life on Earth. Contemporary science has harnessed this knowledge, allowing us to delve deeper into the very fabric of life itself. With the advent of technologies like CRISPR, we are no longer mere spectators in the game of evolution, but active participants, equipped with tools that enable us to edit the very instructions that govern cellular behavior. CRISPR, an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, allows scientists to precisely alter DNA sequences with unparalleled accuracy. This technology, inspired by a natural defense mechanism used by bacteria, permits the targeted editing of genes, providing a revolutionary approach to genetic manipulation. Imagine the possibilities: treating genetic disorders, enhancing crop resilience, and even combating infectious diseases. With the power of CRISPR, we can address some of humanity's most pressing challenges by directly modifying the genes responsible for adverse traits or conditions. Yet, as we navigate this uncharted territory, we must approach CRISPR with caution and responsibility. The ability to edit life comes with ethical considerations and implications that demand our attention. The potential for unintended consequences, such as off-target effects or ecological disruptions, reminds us of the delicate balance we must maintain with nature. As we explore the boundaries of synthetic biology, it is crucial to engage in thoughtful discourse about the moral dimensions of genetic engineering. The promise of CRISPR technology extends beyond mere correction of genetic defects. It opens doors to new avenues in regenerative medicine, where we can potentially repair damaged tissues or organs by modifying the very cells that comprise them. This could revolutionize our approach to healthcare, shifting from reactive treatments to proactive solutions that enhance health and longevity. By understanding and manipulating the genetic underpinnings of life, we are ushering in an era where the possibilities seem boundless, yet grounded in the respect for life that has evolved over millennia. In this unfolding narrative of CRISPR and synthetic biology, we stand on the brink of a new understanding of life itself. We are empowered to rewrite the code of existence, and with that power comes the profound responsibility to ensure our innovations harmonize with the natural world. As we move forward, we must remain vigilant stewards of this knowledge, committed to a vision that embraces both scientific advancement and the preservation of the intricate web of life that surrounds us. As we delve deeper into the realm of DNA and its manipulation, we uncover not only the intricacies of genetic codes, but also the ethical implications that accompany such profound advancements. The ability to edit genes using tools like CRISPR has opened doors to innovative treatments for genetic disorders, agricultural enhancements, and even the potential for de-extinction. Yet with each groundbreaking application, we face questions that challenge our moral compass. What does it mean to alter the very essence of life? As scientists, we are called to navigate these waters with caution, ensuring that our pursuits do not lead to unintended consequences that could disrupt the delicate balance of ecosystems. The intersection of DNA manipulation and evolution presents a unique landscape for ethical discourse. Evolution, a process that has shaped life over millions of years, operates through natural selection and the gradual adaptation of organisms to their environment. When we intervene in this process, we must contemplate the long-term effects of our actions. Are we, in our quest for progress, overriding nature's wisdom? The dialogue surrounding these questions is crucial, as it allows us to reflect upon the responsibilities we hold as custodians of life. Synthetic biology, a field that combines biology with engineering principles, further complicates the ethical landscape. By designing and constructing new biological parts and systems, we are not merely editing existing organisms, we are creating life forms that have never existed before. This capability brings forth innovative solutions to global challenges, such as climate change and food security. However, it also raises significant ethical dilemmas regarding the potential misuse of this technology, the rights of newly created organisms, and the unforeseen consequences of introducing synthetic life into natural ecosystems. As we engage in these discussions, it becomes evident that the ethical considerations in genetic engineering require a multidisciplinary approach. Scientists, ethicists, policymakers, and the public must collaborate to establish guidelines that not only govern the use of biotechnologies, but also reflect societal values and priorities. This collective dialogue can help us navigate the uncertain waters of innovation, ensuring that our technological advancements serve not only humanity, but also the broader tapestry of life on Earth. Ultimately, as we stand on the precipice of a new era in biotechnology, the questions we pose today will shape the future of genetic engineering and its role in society. By fostering an environment of ethical reflection and open dialogue, we can strive to harmonize our scientific endeavors with the reverence for life that has guided us through history. At the core of all living organisms, DNA serves as the blueprint of life, encoding the information necessary for growth, reproduction, and adaptation. Through the intricate processes of evolution, this genetic material has undergone countless changes, gradually shaping the diverse tapestry of life that we observe today. Each species has adapted uniquely to its environment, yet all share the same fundamental building blocks of life, revealing a deep connection among all living beings. As we grapple with the pressing challenges of climate change, it becomes essential to leverage our understanding of DNA and evolution to foster innovative solutions. Synthetic biology emerges as a powerful tool in this context, allowing scientists to design and engineer microorganisms with specific functions. By manipulating the genetic codes of these tiny organisms, we can create strains that are better equipped to absorb carbon dioxide from the atmosphere, effectively transforming them into biological agents of change. Imagine a world where these engineered microbes are deployed in various ecosystems, working in tandem with nature to capture and sequester carbon, helping to mitigate the impacts of climate change. This concept of harnessing the capabilities of microorganisms is not merely a scientific endeavor, it is also a reflection of our relationship with the natural world. By understanding how life has evolved to adapt to its surroundings, we can draw inspiration for our synthetic biology innovations. Furthermore, the ethical implications of these advancements prompt us to consider how we can responsibly navigate the complexities of genetic manipulation, ensuring that our interventions enhance the environment rather than disrupt it. The potential of synthetic biology to combat climate change extends beyond carbon capture. Through targeted genetic modifications, we can also enhance the resilience of crops, enabling them to thrive in changing climates while reducing the need for chemical fertilizers. Such practices not only contribute to food security, but also support sustainable agricultural systems that can coexist harmoniously with the planet. As we continue to explore the intersection of DNA, evolution, and synthetic biology, we must remain vigilant in our commitment to ethical considerations. Every advancement carries the weight of responsibility, and it is imperative that we engage in thoughtful discussions about the implications of our scientific pursuits. By doing so, we can ensure that our efforts to engineer solutions for climate change are grounded in a respect for life and a desire to foster a sustainable future. As we delve deeper into the remarkable world of bioremediation, we are reminded of the intricate relationship between DNA, evolution, and synthetic biology. Bioremediation refers to the use of living organisms, often bacteria or plants, to degrade environmental pollutants and restore ecosystems. Through the lens of synthetic biology, scientists are now able to engineer these organisms at the genetic level, enhancing their ability to break down harmful substances such as heavy metals, petroleum products, and pesticides. This intersection of advanced genetic manipulation and natural selection opens up new avenues for addressing pollution, allowing us to harness the power of evolution in a controlled manner. By tailoring the genetic makeup of these organisms, we can optimize their efficiency and effectiveness in detoxifying contaminated environments. The process of bioremediation is a beautiful testament to the resilience of life and the potential for synthetic biology to contribute positively to our planet. When we consider how organisms adapt and evolve over time, we recognize that nature has already been perfecting these processes for millions of years. However, with the rapid pace of industrialization and environmental degradation, natural mechanisms alone may not suffice. Through synthetic biology, we can accelerate evolutionary processes, creating organisms that are not only capable of surviving in harsh conditions, but also excelling at breaking down pollutants. This engineered resilience not only restores ecosystems, but also emphasizes the responsibility we hold in using our knowledge wisely. As we harness the capabilities of synthetic organisms for bioremediation, ethical considerations must remain at the forefront of our endeavors. The manipulation of DNA, while offering unprecedented opportunities for environmental restoration, also raises questions about the unintended consequences of releasing engineered species into the wild. Understanding the ecological balance is essential, as these organisms could interact with native species in unforeseen ways. Therefore, rigorous testing and monitoring are crucial components of any bioremediation strategy. Our commitment to ethical stewardship ensures that while we innovate, we do so with an awareness of the delicate web of life that sustains us. Furthermore, the potential of synthetic biology extends beyond mere pollution cleanup. It embodies a vision for a sustainable future where engineered organisms can be designed not only to remediate, but to also thrive in various ecosystems, enriching biodiversity while mitigating human impact. This transformative approach invites us to rethink our relationship with nature, shifting our perspective from one of dominance to one of collaboration. By integrating synthetic biology into our environmental strategies, we pave the way for a new era of restoration, where humans and nature can work hand in hand towards a common goal. As we contemplate the profound implications of bioremediation and synthetic biology, it becomes evident that our journey is only beginning. The fusion of science and ethics in our approach will shape the outcomes of our interventions on the planet. The potential to engineer life for the betterment of our environment is both exhilarating and daunting. With every advancement, we must remain vigilant, ensuring that our innovations serve the greater good while honoring the intricate dance of life that has evolved over millennia. The intricate structure of DNA serves as the blueprint for life, encoding the vast array of biological information that governs the development and function of organisms. Through the lens of evolution, we understand that the genetic variations occurring over generations have resulted in the remarkable diversity we observe today. Yet, with the advent of synthetic biology, we are presented with the opportunity to transcend traditional evolutionary mechanisms. By harnessing our understanding of DNA, scientists can create genetically modified organisms that are tailored to withstand environmental stressors, enhance nutritional content, and ultimately contribute to more sustainable food production. As we delve deeper into the realm of synthetic biology, we encounter the concept of lab-grown meat, a revolutionary advancement aimed at addressing the environmental impact of livestock farming. This innovative approach utilizes cellular agriculture to produce animal cells in controlled environments, eliminating the need for traditional animal husbandry. By replicating the natural growth processes of cells, we can cultivate meat that is not only ethically produced, but also significantly reduces greenhouse gas emissions and land use. This shift towards lab-grown meat epitomizes the potential of science to innovate solutions for pressing global challenges while maintaining a respect for the ecological balance that has evolved over millennia. Furthermore, the role of synthetic biology extends beyond merely enhancing food production. It also invites us to reconsider our relationship with nature. The engineering of organisms raises essential ethical questions about the boundaries of manipulation and our responsibilities as stewards of the planet. As we embrace these transformative technologies, it is crucial to foster a dialogue around the implications of genetic modification and the balance between human ingenuity and natural systems. Only through thoughtful contemplation can we ensure that our advancements lead to a harmonious coexistence with the environment, honoring both the complexity of nature and our aspirations for a sustainable future. The integration of synthetic biology into food production also presents a unique opportunity to address food security challenges faced by a growing global population. By optimizing crop yields through genetically engineered plants that are resistant to pests and diseases, we can produce more food using fewer resources. This not only enhances food availability, but also reduces the environmental footprint of agriculture, aligning with our collective goal of preserving the planet's resources for future generations. As we explore these possibilities, we must remain attuned to the lessons of evolution, ensuring that our innovations are guided by principles of sustainability and responsibility. In this exciting landscape of possibilities, the convergence of DNA science, evolution, and synthetic biology opens up new pathways for the future of food production. As we continue to unravel the complexities of genetic manipulation and its applications, we find ourselves at a pivotal moment in history, where the choices we make will leave lasting impacts on our environment and society. It is essential to navigate this terrain with foresight and care, recognizing that our endeavors in synthetic biology must always be anchored in the principles of ethical responsibility and ecological harmony. With each step forward, we are not only shaping the future of food, but also redefining our role within the tapestry of life on Earth. At the heart of synthetic biology lies our understanding of DNA, the very blueprint of life. This intricate molecule carries the instructions that dictate the functioning of all living organisms. By meticulously unraveling the sequences of DNA, scientists have begun to harness its potential, paving the way for groundbreaking innovations in medicine. When we consider evolution, we see how life has adapted over millions of years through natural selection, leading to an astonishing diversity of forms and functions. Now, with synthetic biology, we can mimic these natural processes, designing organisms and systems that not only address existing health challenges, but also preemptively combat potential threats. The advancements in vaccine development exemplify the transformative potential of synthetic biology. By utilizing engineered viral components, researchers are able to craft vaccines that teach the immune system to recognize and combat pathogens more effectively. This tailored approach is grounded in our evolving understanding of genetics and immune response, allowing for the rapid development of vaccines against emerging diseases. Furthermore, therapies that employ synthetic biology techniques, such as gene editing, hold promise for treating genetic disorders at their source. By correcting mutations in an individual's DNA, we can offer patients a chance at a healthier life, representing a monumental shift in our approach to medicine. As we delve deeper into the realm of synthetic biology, the ethical implications of our advancements become increasingly salient. It is crucial to maintain a dialogue about the societal impacts of our innovations, ensuring that the benefits of synthetic biology are accessible to all. The technology has the potential to revolutionize healthcare, but we must approach its application with a sense of caution and responsibility. By fostering interdisciplinary collaboration among scientists, ethicists, and policymakers, we can create a framework that guides the responsible use of synthetic biology in medicine. This collaborative effort will not only enhance the efficacy of our medical advancements, but also safeguard the integrity of the ecosystems we inhabit. The exploration of synthetic biology also invites us to rethink our relationship with nature. Rather than viewing ourselves as separate from the biological systems that surround us, we are increasingly recognizing our role as stewards of these intricate networks. As we manipulate DNA to develop new medical therapies, we must remain aware of the interconnectedness of life. With each engineered organism or therapeutic intervention, we are engaging in a dialogue with the natural world, one that demands respect and careful consideration. This harmonious approach not only enhances our medical capabilities, but also reinforces the importance of preserving biodiversity as we forge ahead into the future of healthcare. In this unfolding narrative, the practical applications of synthetic biology serve as a bridge between scientific discovery and societal benefit. By engaging with real-world problems through innovative solutions, we can illuminate pathways to a sustainable future. Yet, as we navigate this terrain, it remains crucial to balance ambition with ethical considerations, ensuring that our endeavors align with the broader goals of preserving life and fostering harmony within the biosphere. As we reflect on these principles, we prepare ourselves to explore the intricate dance between technology and nature that defines our ongoing journey into the future. Gene drives represent a revolutionary advancement in our understanding of genetic inheritance. By utilizing engineered DNA sequences that can propagate through generations at an accelerated rate, scientists are able to establish genetic traits within wild populations more rapidly than traditional methods would allow. This capability raises profound questions about the manipulation of life and the potential consequences of altering genetic dynamics within ecosystems. As we consider the implications of gene drives, we are reminded of the delicate balance that sustains biodiversity. The introduction of a modified gene into a population not only affects the intended target species, but can also have cascading effects on the entire ecosystem. The potential applications of gene drives are immense and span various fields, including agriculture, pest control, and even conservation efforts. For instance, gene drives could be employed to reduce populations of invasive species that threaten native flora and fauna, thereby aiding in the restoration of ecological balance. However, the ethical considerations of such interventions loom large. As stewards of the environment, we must weigh the benefits of gene drives against the risks of unintended consequences that might arise from disrupting established relationships within ecosystems. Each action taken to engineer life carries with it the responsibility to ensure that we do not inadvertently cause harm to the very systems we seek to protect. Moreover, the spread of genetic changes facilitated by gene drives challenges our understanding of evolution itself. Natural selection and adaptation have long been governed by the slow, gradual processes of mutation and environmental pressures. With gene drives, we introduce a new paradigm where human intention can rapidly influence the course of evolution. This acceleration raises questions about the natural resilience of ecosystems and the capacity of organisms to adapt to sudden changes. As we venture into this new territory, we must consider how our interventions align with the principles of evolutionary biology and the long-term implications for species survival. As we explore the frontiers of synthetic biology, it is essential to maintain a holistic perspective on the implications of our innovations. The integration of gene drives into our biological toolkit should be approached with caution and foresight. We must engage in open dialogues about the potential risks and rewards, involving not just scientists, but also ethicists, ecologists, and the broader public. By fostering a collaborative approach, we can better navigate the complexities of genetic manipulation while honoring the intricate web of life that sustains us. In doing so, we open the door to a future where technology and nature coexist in a balanced, respectful harmony. In this evolving narrative, the role of gene drives serves as a poignant reminder of our interconnectedness with the natural world. As we harness the power of synthetic biology to address pressing global challenges, we must remain vigilant stewards of the ecosystems we inhabit. The dialogue surrounding gene drives invites us to reflect not only on our technological capabilities, but also on the ethical frameworks that guide our actions. As we look ahead, it becomes increasingly clear that the choices we make today will shape the legacy we leave for future generations. At the core of life lies the intricate molecule known as DNA, the blueprint of all living organisms. This double helix structure, composed of nucleotides, encodes the information necessary for the development, functioning, and reproduction of life forms. Through the processes of evolution, driven by natural selection, this genetic material has been shaped and reshaped over millennia, leading to the remarkable diversity of life we witness today. Each mutation, whether advantageous, neutral, or detrimental, contributes to the tapestry of life, influencing how species adapt to their environments. As we delve deeper into the mechanisms of DNA, we uncover not only the pathways of evolution, but also the potential for human intervention through synthetic biology, which seeks to redesign biological systems for our benefit. Synthetic biology represents a powerful convergence of disciplines, merging biology with engineering principles to create novel organisms or modify existing ones. This field opens up a plethora of possibilities, from developing sustainable biofuels to engineering microbes that can produce pharmaceuticals. By understanding the genetic code, scientists can manipulate DNA sequences to enhance desired traits or suppress unwanted ones, sparking discussions about the implications of such power. The ability to design life forms or alter ecosystems brings forth ethical considerations that challenge our understanding of what it means to be a steward of the earth. The intersection of DNA, evolution, and synthetic biology not only illuminates how life adapts, but also empowers us to influence these processes, leading to a profound responsibility to consider the ramifications of our innovations. As we explore the potential of synthetic biology, we find ourselves at a crossroads of innovation and ethics. The ability to edit genes, particularly through techniques like CRISPR, allows us to make precise changes in the DNA of organisms, which can lead to enhanced agricultural yields or the eradication of diseases. However, with this technological prowess comes the necessity of reflection on our choices and their consequences. Each alteration we make resonates through the web of life, potentially affecting not only the targeted species, but also the broader ecosystems they inhabit. It is imperative to integrate ethical considerations into our scientific explorations, ensuring that our actions honor the delicate balance of nature, thus fostering a harmonious coexistence with all living beings. The dialogue surrounding DNA and synthetic biology also extends to the philosophical realms of identity and agency. As we gain the ability to modify life itself, questions arise about the nature of evolution and whether we are continuing a natural process or stepping outside of it. Are we the architect of a new era of biological existence, or are we merely participants in an ongoing evolutionary journey? This contemplation encourages a broader understanding of our role within the ecosystem, highlighting the importance of humility and respect for the natural world. By recognizing our interconnectedness, we can approach synthetic biology not just as a tool, but as a profound opportunity to enhance life while preserving the integrity of the systems that sustain us. As we stand at this intersection of DNA, evolution, and synthetic biology, we must embrace a vision that harmonizes scientific advancement with ecological stewardship. This requires a commitment to ongoing dialogue among scientists, ethicists, and the public, fostering a culture of transparency and inclusivity in decision-making processes. The potential of synthetic biology is vast, yet it is intertwined with the delicate fabric of life that has evolved over eons. By remaining attuned to the implications of our innovations, we can steer our trajectory toward a future where technology and nature coexist in a balanced relationship. As we navigate this complex landscape, let us remain vigilant in our choices, ensuring that the legacy we forge is one of wisdom and respect for the intricate web of life that surrounds us. Bioinformatics plays a pivotal role in unraveling the complexities of DNA, serving as a bridge that connects raw genetic information with the profound insights of evolutionary biology. In an era where we are inundated with an overwhelming amount of genetic data, bioinformatics provides the necessary tools and methodologies to analyze and interpret this information efficiently. By employing algorithms and computational models, researchers can identify patterns and relationships within the vast sea of genetic sequences, shedding light on how traits are inherited and how organisms adapt to their environments over time. This analytical framework not only enhances our understanding of individual species, but also illuminates the intricate connections that bind all life forms together in the grand tapestry of evolution. The applications of bioinformatics extend far beyond merely cataloging genetic sequences. They empower scientists to explore evolutionary pathways and the mechanisms of natural selection. By comparing genomes across different species, bioinformatics allows us to trace the lineage of specific genes and uncover the evolutionary pressures that have shaped their development. This comparative analysis not only deepens our understanding of the history of life on Earth, but also provides insights into the genetic basis of diseases and the resilience of species in the face of environmental changes. As we delve deeper into the genetic blueprints of organisms, bioinformatics serves as a guiding light, illuminating the pathways that lead from ancient ancestors to the rich diversity of life we witness today. Furthermore, bioinformatics is instrumental in the realm of synthetic biology, where the boundaries of natural systems are increasingly blurred. By leveraging the vast datasets generated through genomic research, scientists can design and engineer new biological systems with precision. This engineering aspect allows researchers to create synthetic organisms that carry out specific functions, such as producing renewable energy or synthesizing pharmaceuticals. However, this powerful capability necessitates a careful consideration of ethical implications and ecological impacts as we tread into territories where nature and technology intersect. Through bioinformatics, we can assess the potential consequences of these innovations, ensuring that synthetic biology aligns with our commitment to ecological stewardship and the preservation of biodiversity. In this age of rapid technological advancement, the role of bioinformatics becomes even more critical as we seek to unravel the mysteries of life at the molecular level. Integration of artificial intelligence and machine learning into bioinformatics is opening new frontiers, enabling researchers to predict genetic interactions and model evolutionary scenarios with greater accuracy. Such advancements not only enhance our understanding of fundamental biological processes, but also facilitate the development of personalized medicine, where treatments can be tailored to an individual's genetic makeup. As we harness these powerful tools, we must remain aware of the responsibility that accompanies such knowledge, ensuring that our scientific pursuits are guided by ethical considerations and a commitment to the greater good. As we continue to explore the potential of bioinformatics, we find ourselves in a unique position to not only understand the past, but also shape the future of life on our planet. The insights gained from bioinformatics can inform conservation efforts, guiding us in the protection of endangered species and the restoration of ecosystems that have been disrupted by human activity. By understanding the genetic diversity within populations, we can develop strategies to enhance resilience against changing climates and environmental pressures. It is here that the synergy between bioinformatics, evolutionary theory, and synthetic biology becomes profoundly significant, offering a pathway toward a more harmonious coexistence with nature. At the heart of our understanding of evolution lies DNA, the intricate blueprint of life that encodes the genetic information necessary for the development and functioning of all living organisms. Recent advancements in genomic sequencing have illuminated the vast complexity of this molecular structure, revealing not only the similarities that bind different species together, but also the unique variations that contribute to their diversity. By examining the genetic code across species, evolutionary biologists are uncovering the pathways through which life has adapted and evolved over millions of years. Each discovery adds a piece to the puzzle of how organisms respond to their environments, survive challenges, and thrive. Furthermore, the study of DNA is increasingly intertwined with synthetic biology, a field that seeks to manipulate the genetic material of organisms to create new biological systems and functions. This innovative approach allows scientists to not only replicate natural processes, but also to engineer novel solutions to pressing issues, such as food security and disease management. By harnessing the principles of evolution, researchers can design organisms that are better suited to withstand environmental stresses or produce valuable compounds. Such capabilities present a remarkable opportunity to rethink our relationship with nature, moving toward a future where human ingenuity and natural processes coexist harmoniously. As we delve deeper into the genetic underpinnings of evolution, we also confront ethical considerations that arise from our ability to manipulate life itself. The potential consequences of synthetic biology extend beyond the laboratory, prompting discussions about the ecological impact of genetically modified organisms and the moral implications of altering life forms. It is essential that these conversations are grounded in a robust understanding of evolutionary principles, ensuring that our innovations are informed by the complexities of life's history. By integrating evolutionary biology with ethical frameworks, we can navigate the challenges posed by synthetic biology and ensure that our actions align with the greater good of the planet. The exploration of evolutionary biology not only enriches our understanding of the past, but also provides a compass for navigating the future of biodiversity. By studying the genetic variations that have emerged over time, we can identify the traits that confer resilience and adaptability in changing environments. This knowledge is invaluable for conservation efforts as it allows us to prioritize the protection of species that hold significant ecological roles or genetic diversity vital for future adaptation. In this way, the insights gleaned from evolutionary research act as a guiding light, illuminating the path towards sustainable interactions with our ecosystems. In the realm of synthetic biology, the possibilities for innovation are boundless, yet they are intricately woven into the fabric of evolutionary history. As we engineer new life forms, we must remain mindful of the lessons learned from the natural world, ensuring that our creations do not disrupt the delicate balance of ecosystems. The interplay between synthetic biology and evolutionary theory fosters a deeper respect for the processes that have shaped life on Earth, encouraging us to create responsibly and sustainably. As we stand at the intersection of these fields, the potential for transformative change becomes evident, urging us to explore the uncharted territories of life with both curiosity and caution. The evolution of antibiotic resistance in bacteria serves as a profound illustration of how evolution operates at the molecular level through the mechanisms of DNA. When antibiotics are introduced into a bacterial population, they create a selective pressure that favors the survival of those individuals with mutations or genetic traits that confer resistance. This process, driven by random genetic mutations and the rapid reproduction of bacteria, unfolds in a remarkably short timeframe, often over the course of just a few generations. The DNA of these microorganisms acts as both the blueprint of life and the record of their evolutionary journey, capturing the changes that allow them to adapt to new challenges. By studying the genetic changes associated with antibiotic resistance, scientists gain insight into the principles of natural selection, demonstrating that evolution is not just a historical narrative, but a dynamic process occurring in real time. As we delve deeper into the mechanics of antibiotic resistance, we uncover the intricate relationship between evolution, DNA, and the implications for human health. Each resistant strain of bacteria tells a story of survival, revealing how genetic information can be exchanged between different species through processes like horizontal gene transfer. This exchange accelerates the spread of resistance traits, complicating our efforts to combat bacterial infections. The understanding of these evolutionary dynamics is crucial for developing new antibiotics and alternative therapies, enabling us to stay ahead of rapidly evolving pathogens. Moreover, this knowledge serves as a reminder of our role in shaping the microbial world, urging us to use antibiotics judiciously to mitigate the emergence of resistance. The implications of antibiotic resistance extend beyond individual health, influencing public health policies and ecological balance. The rise of resistant bacteria necessitates a comprehensive approach that encompasses not only medical intervention, but also a deep understanding of the evolutionary principles that govern microbial life. This understanding fosters a holistic perspective, encouraging collaboration between biologists, medical professionals, and policymakers. As we confront the challenges posed by antibiotic resistance, we must acknowledge the interconnectedness of life on Earth, recognizing that our actions can have far-reaching consequences for all organisms. The synergy between synthetic biology and our comprehension of evolutionary processes offers promising avenues for innovation, where engineered solutions may one day coexist with natural ecosystems in harmony. In this context, synthetic biology emerges as a powerful tool that can complement our understanding of evolution while addressing the pressing issue of antibiotic resistance. By harnessing genetic engineering techniques, scientists can design novel microbes capable of outcompeting resistant strains or producing new antimicrobial compounds. This approach not only seeks to mitigate the threat of resistant bacteria, but also highlights the potential of synthetic organisms to contribute positively to ecological systems. However, as we venture into this realm of creation, it is imperative to tread with caution, ensuring that our engineered solutions are safe and effective within the ecosystem. The evolution of antibiotic resistance remains a poignant reminder of the power of adaptation, urging us to consider the ethical dimensions of our innovations while honoring the wisdom of nature's own evolutionary strategies. As we reflect on the lessons learned from studying antibiotic resistance, we find ourselves at a critical juncture where science, ethics, and the future of life intersect. The ongoing battle against resistant bacteria is not merely a medical challenge, but a call to engage deeply with the principles of evolution and the role of DNA. In navigating this complex landscape, we are reminded that the stories written in the genetic code of organisms are not just tales of survival, but also reflections of our responsibility to maintain the delicate balance of life. With each new discovery, we are invited to explore the potential of synthetic biology, guided by a profound respect for the intricate dance of evolution that has shaped the diversity of life on our planet. As we delve deeper into the realm of synthetic evolution, we uncover the profound potential embedded within the very structure of DNA. The double helix is not merely a blueprint for life, it is a dynamic repository of information that has been honed over billions of years through natural selection. Each gene, a testament to the adaptive strategies that organisms have employed to thrive in their environments, offers a glimpse into the mechanics of evolution itself. By harnessing the tools of synthetic biology, scientists are beginning to rewrite these genetic narratives, engineering organisms that possess the ability to adapt to rapidly changing conditions. This approach provides a powerful means to address some of the most pressing challenges of our time, such as climate change and resource scarcity. The process of synthetic evolution invites us to reimagine the boundaries of biological possibility. Through techniques such as CRISPR and gene synthesis, we can not only edit existing organisms, but also create novel life forms that may exhibit entirely new traits and functions. The implications of these advancements are vast. Engineered microbes, for instance, could be designed to metabolize pollutants or produce renewable energy, thereby enhancing ecosystem resilience. Yet, with this incredible power comes a weighty ethical responsibility. As we manipulate the very fabric of life, we must remain vigilant stewards, ensuring that our creations do not disrupt the balance of ecosystems or lead to unintended consequences. The concept of synthetic evolution also encourages us to reflect on the interconnectedness of life. Every organism, from the simplest bacteria to complex mammals, is part of an intricate web of relationships, shaped by evolutionary forces. By engineering organisms that can better adapt to their environments, we are not merely altering individual species, we are influencing the entire ecological landscape. This perspective urges us to consider the long-term effects of our interventions, fostering a mindset that values harmony and sustainability in the biosphere. It is through this lens that we can appreciate the delicate dance of evolution, guiding our exploration of synthetic biology with humility and foresight. Looking ahead, the future of synthetic evolution holds immense promise, yet it also beckons us to engage with the ethical implications of our creations. As we engineer organisms to thrive in a world of uncertainty, we must cultivate a collaborative dialogue among scientists, ethicists, and the public. This conversation will be vital in shaping the frameworks that govern synthetic biology, ensuring that our innovations align with our values and the well-being of our planet. By fostering transparency and inclusivity in our approach, we can navigate the complexities of this new frontier with wisdom and care, paving the way for a future where engineered organisms coexist harmoniously with the natural world. As we stand at this intersection of technology and nature, the journey into synthetic evolution is just beginning. The potential to create organisms that can adapt and thrive in diverse environments offers a pathway to resilience, sparking hope for a sustainable future. With each step forward, we are reminded that we are not separate from nature, but rather integral to its ongoing story. As we continue our exploration, we must remain attuned to the lessons of evolution, allowing them to guide us in our quest to harness the power of synthetic biology for the greater good. As we delve deeper into the realm of genetic engineering, it becomes increasingly necessary to consider the public's perception of these transformative technologies. The human connection to genetics is profound, as we recognize that our very essence, encapsulated in DNA, forms the foundation of life itself. Yet, this intricate tapestry of life is often viewed through the lens of apprehension and misunderstanding. The media, educational institutions, and scientific communities play pivotal roles in shaping public opinion, influencing how we perceive the implications of genetic modification. By fostering a dialogue that emphasizes transparency and education, we can begin to bridge the gap between scientific knowledge and public sentiment. Understanding the complexities of public opinion is vital for guiding the responsible advancement of genetic engineering. As we explore the potential of synthetic biology, we must acknowledge the ethical concerns and societal impacts that accompany these innovations. Issues such as food security, environmental sustainability, and healthcare can evoke strong emotions and fears among the public. By actively engaging communities in conversations about the benefits and risks associated with genetic engineering, we can cultivate a more informed populace. This approach not only empowers individuals, but also encourages them to participate in shaping policies that govern the use of these technologies. Moreover, the evolution of public perception is influenced by cultural narratives and historical context. Throughout history, humanity has grappled with the ramifications of scientific advancements, often oscillating between awe and skepticism. The legacy of past scientific endeavors, such as the introduction of antibiotics and vaccines, serves as a reminder of the potential for both great benefit and unforeseen consequences. As we navigate the complexities of genetic engineering, it is imperative to consider these historical lessons, allowing us to approach new technologies with both optimism and caution. Through this lens, we can foster a culture that embraces innovation while remaining vigilant about ethical considerations and societal effects. In this evolving landscape, scientists and policymakers must collaborate to create frameworks that reflect the values and concerns of society. By prioritizing public engagement, we can demystify genetic engineering and promote a more nuanced understanding of its potential. Initiatives such as community forums, educational outreach, and transparent research practices can empower individuals to voice their opinions and contribute to the discourse surrounding genetic technologies. Ultimately, cultivating an informed and engaged public will facilitate the responsible integration of genetic engineering into our lives, ensuring that these advancements serve humanity's best interests. As we reflect on the intricate relationship between public perception and genetic engineering, we recognize the importance of empathy and understanding in this dialogue. The journey toward a future enriched by synthetic biology is not solely a scientific endeavor. It is a collaborative effort that requires the input and perspectives of diverse communities. By embracing this complexity, we can build a foundation of trust that enables us to harness the power of genetic engineering for the greater good. With each conversation, we draw closer to realizing a future where technology and nature coexist harmoniously, guided by the wisdom of our shared experiences. As we stand at the intersection of DNA, evolution, and synthetic biology, we find ourselves in the unique position to shape the future of life itself. The molecular blueprint of DNA has long fascinated scientists, revealing the intricate codes that dictate the functions and traits of all living organisms. Through the lens of evolution, we understand that these codes are not static. They evolve over generations, adapting to ever-changing environments and circumstances. This dynamic nature of DNA serves as a reminder of the resilience and creativity inherent in life. In this context, synthetic biology emerges as a powerful tool, allowing us to manipulate these fundamental codes to solve pressing challenges, from disease prevention to sustainable agriculture. However, with great power comes great responsibility. As we harness the capabilities of synthetic biology, we must remain vigilant and thoughtful about the ethical implications of our actions. The potential to edit genomes and create new life forms raises important questions about the boundaries of our interventions in nature. It is crucial that we engage with a diverse array of voices in these discussions, ensuring that the benefits of innovation are equitably shared and that the risks are carefully managed. By fostering a culture of transparency and collaboration, we can establish frameworks that prioritize the well-being of both humanity and the ecosystems we inhabit. As we look ahead, the promise of synthetic biology is not merely about technological advancement. it is about reimagining our relationship with the natural world. This journey invites us to reflect on our values and aspirations as we consider how to integrate cutting-edge science with the principles of sustainability and respect for life. By embracing the complexity of this endeavor, we can envision a future where innovation is guided by ethical considerations, ensuring that our advancements contribute positively to the planet. Ultimately, the path forward requires a commitment to continuous learning and adaptation. As we deepen our understanding of DNA and the mechanisms of evolution, we will better equip ourselves to navigate the challenges and opportunities presented by synthetic biology. The dialogue surrounding these topics is ongoing, and it is through collective engagement that we can foster a more informed and compassionate society. In this shared journey, we come together to embrace the unknown, guided by curiosity and a sense of responsibility toward future generations. In conclusion, as we embrace the future shaped by DNA, evolution, and synthetic biology, we are reminded of the profound interconnectedness of all life. This intricate tapestry of genetic information and evolutionary processes not only defines who we are, but also illuminates the path forward. By honoring our shared responsibility to nurture this relationship, we can cultivate a future where scientific innovation harmonizes with ethical stewardship, ultimately enriching the tapestry of life for all. The journey of scientific discovery is often illuminated by the contributions of countless individuals whose collective efforts have propelled our understanding of complex phenomena. When we think of DNA, we often recall the groundbreaking work of James Watson and Francis Crick, whose discovery of the double helix structure in 1953 has paved the way for modern genetics. Yet, it is crucial to recognize that this momentous achievement was built upon the foundations laid by many others, including Rosalind Franklin, whose pioneering use of X-ray diffraction provided essential insights into the molecular architecture of DNA. Each advancement in our understanding of evolution, from Charles Darwin's theory of natural selection to the modern synthesis incorporating genetics, exemplifies the collaborative spirit of science, where ideas evolve and flourish through shared knowledge and rigorous debate. As we delve deeper into the realm of synthetic biology, we find ourselves standing on the shoulders of giants, drawing from the wisdom and research of those who have come before us. This interdisciplinary field merges biology with engineering, allowing scientists to redesign organisms for beneficial purposes, such as producing biofuels or creating new medical therapies. The groundbreaking work of pioneers such as George Church and Jennifer Doudna has opened new pathways in genetic editing and synthetic genome design, leading to transformative possibilities. Their contributions remind us that science is not merely a collection of individual achievements, but rather a tapestry woven from collaboration, creativity, and the unwavering pursuit of knowledge. Recognizing the collaborative nature of scientific inquiry allows us to appreciate the intricate web of relationships that underpins every discovery. Each scientist, researcher, and student plays a vital role in the progression of knowledge, sharing insights, challenges, and triumphs along the way. The work of institutions and research communities foster environments where ideas can flourish, leading to innovations that can redefine our understanding of life itself. As we explore the intersections of DNA, evolution and synthetic biology, we must acknowledge that our advancements are the result of a shared commitment to inquiry, where diverse perspectives and expertise converge to illuminate the path forward. As we stand at the crossroads of biology and technology, it becomes increasingly clear that the future of science lies in continued collaboration and mutual respect among researchers from various disciplines. The potential of synthetic biology to address global challenges, from climate change to healthcare, depends on the collective intelligence of scientists working together, sharing knowledge and resources. This collaborative ethos is essential, as it fosters an environment where innovative ideas can be born, nurtured, and brought to fruition. The story of scientific discovery is one of unity in diversity, reminding us that every contribution, no matter how small, is part of a greater narrative that shapes the world we inhabit. As we continue our exploration into the intricacies of DNA and evolution, we find that the interplay between natural processes and human ingenuity opens up a realm of possibilities previously thought unattainable. The ongoing advances in synthetic biology not only redefine our understanding of life, but also pose profound ethical questions about our role as stewards of the natural world. This evolving dialogue encourages us to reflect on how we can responsibly harness the power of science to create a sustainable future grounded in respect for the intricate relationships that bind all living organisms. DNA, the fundamental building block of life, serves as a remarkable repository of information that guides the development and functioning of all living organisms. Each organism's unique DNA sequence encodes the instructions necessary for growth, reproduction, and adaptation, showcasing the beauty of evolution in action. As species navigate the challenges posed by their environments, they undergo gradual changes, mutations and variations that can lead to significant evolutionary advancements over generations. This delicate process, driven by natural selection, highlights the interconnectedness of all life forms and the intricate dance of survival that unfolds on our planet. As we delve deeper into the concept of evolution, we recognize that it is not merely a historical account of life on Earth, but an ongoing narrative that shapes our present and future. The understanding of evolutionary biology allows us to appreciate the complexities of ecosystems and the adaptive strategies that organisms employ to thrive. However, with the advent of synthetic biology, we find ourselves at a crossroads where the boundaries between natural evolution and human intervention blur. Synthetic biology empowers us to manipulate genetic codes, enabling the design of new organisms and the enhancement of existing ones, thus raising essential questions about our ethical responsibilities in this brave new world. The innovations arising from synthetic biology hold immense potential for addressing pressing global challenges, from food security to disease prevention. By understanding and applying the principles of DNA and evolution, scientists can engineer microorganisms to produce sustainable biofuels, create crops resistant to climate change, and develop therapies that target specific genetic disorders. Yet, these advancements compel us to consider the long-term implications of our actions on the natural world. As we unlock the secrets of life through genetic engineering, we must remain vigilant stewards, ensuring that our pursuit of knowledge aligns with the preservation of biodiversity and ecological balance. The dialogue surrounding DNA, evolution, and synthetic biology invites us to reflect on our role as custodians of the planet. Every innovation brings with it a responsibility to consider not only the scientific possibilities, but also the ethical dimensions that accompany such power. It is through thoughtful communication and collaboration within the scientific community and society at large that we can navigate these complexities. By honoring the contributions of researchers, ethicists, and educators, we foster a culture of transparency and inclusivity in scientific discourse, ensuring that the journey toward a sustainable future is one that respects the interconnectedness of all life forms. As we conclude this exploration, let us carry forward the understanding that the marvels of DNA and the principles of evolution are not isolated phenomena, but integral to the fabric of life itself. The advances in synthetic biology offer profound opportunities for innovation, yet they also remind us of our duty to tread lightly upon the earth. In honoring the creators of knowledge and the legacies of scientific inquiry, we reinforce the importance of communication in shaping a future that is both informed and compassionate. Together, through shared understanding and respect for the natural world, we can foster a harmonious relationship with the wonders of life that surround us.